Round Tower is a 10-year-old solution provider based out of Cincinnati, Ohio, serving the eastern U.S. We've had really explosive growth, especially over the last 5 years, and a good bit of that is because as we work to craft solutions, we bring deep industry expertise and experts to the table to make sure we're taking our customers where they need to go. We have 5 focus areas: data center infrastructure, mobility and collaboration, orchestration and automation to help customers get to the cloud, whether it be a private cloud on their premise or the public cloud or a hybrid of the two, DevOps, and data analytics. Data analytics in 3 areas: data integration, assisting customers getting to the right level of data quality, and also analytics and analytics solutions in a couple of different areas. Our next speaker is the Chief Executive Officer and Senior Architect for NuRelm. Please welcome Sam Shaaban.
How about this? I sound more impressive now? All right, I think I have to be a little bit quieter now. So that's something that we do a lot of, and we are working, for example, right now on a fairly big project with MailChimp, integrating their new e-commerce functionality that they've rolled out with a particular online commerce platform. We're actually building a tool to help do that, a product that we're hoping to roll out this month called FlowLink. So we do a lot of that work, and that feels similar to the story I'm about to tell you about, in that there is a lot of data that has to be analyzed and kind of wrangled and put into place. A second area that we work is that of mobile application development for sort of the innovation, the generalized innovation space, which is a really interesting area. We're doing some work for the Allegheny Trail Alliance, for example, helping people navigate the Great Allegheny Passage, which is a trail system from Pittsburgh to Washington, using an app that we've helped them build. We're working with the Pennsylvania Resource Commission, helping them with a mobile app connect for-profit businesses that want to get rid of furniture to nonprofits who need furniture. But the third area that we spent a lot of time working in lately, let me try to get to my first slide here. Let's see. So do I need to, there we go. All right, so this is our slide. And the third area is one that's pretty interesting. We've been working in it for a few years now in kind of this research, healthcare research related area. A couple of years ago, maybe a few years ago, the Clinical and Translational Science Institute at Pitt had a competition called PINCH, the Pittsburgh Innovation Challenge. And the idea was they thought that it would be a great idea to get researchers hooked up with business people in such a way that really risky ideas could have some funding put into those ideas. So we thought about that and we thought, oh, really risky ideas. How about this? How about if we present the idea for a computer vision system that uses machine learning to enable you via a mobile application to take a picture of the food that you're eating and guesstimate the carbs in that food so that type 1 diabetics can then estimate how much they need to take of their medication. Talk about an ambitious idea. So we got a couple of really, a pretty good team of researchers together. A gentleman from CMU, computer vision researcher, a couple of really interesting people from Pitt and Children's Hospital that were in the adolescent medicine space. I'm a software developer. We have a world famous epidemiologist, who's actually my wife. She was getting her PhD at the time. And we put a proposal together and we pitched this thing and we got to the very last stage. And I remember we got to the last presentation and I remember one of the judges saying, that's a really cool idea. Do you guys think you can really pull that off? And I said, yeah. So I'm not here to present that idea because we didn't win. However, I do think we could have done that idea. But that opened up a great world of relationships and opportunities. And since that time, we've worked with a couple of the researchers on that same team on a few different projects. One was a behavioral mobile health intervention for young adults. We're working right now on an app to help gather data from 1,400 participants in a study. And we've gotten the opportunity via the relationships there to present our proposal to do some work with the Pittsburgh Health Data Alliance on one of their first 6 projects. And that's the project I'm going to tell you about today. It's a super fascinating area, gives us a lot of opportunity to learn new things while doing the things that we do best, developing apps. So what's that project? There's a lot of interest right now in these words that get used in a way where they all sort of commingle. But let's talk about personalized healthcare as healthcare that takes the patient into account more than just kind of general procedures. You have X, we prescribe Y. We've all heard about treatments and therapies that are not as effective as they should be, or not as effective for one patient as they are with another patient. And I think the dream is really to be able to take biomarkers from a particular person and really specify what medication is going to work. So, if you need an antidepressant, figure out which one is going to work for you, because many of them don't work for the people that they're prescribed to. And I think really the golden egg there would be the thing that we all think about, I think a lot, is this idea of precision oncology, where we can look at somebody's genome and we can figure out how to treat a cancer in such a way that there's a pretty high chance, even if it's an aggressive cancer, that they'll be cured. However, this lady knows a secret, precision oncology isn't real yet, or maybe I should say it's not proven yet. There was an article a few months ago in 2016 in Nature. It was a perspective article, and I think the title was "The Precision Oncology Illusion." And in that article, an oncologist pointed out a couple of things. First of all, few patients have been paired with targeted drugs based on genetic data. It's happened, but it's not something that happens very often, and there aren't a lot of cases where that's happened. Of those cases, trials that have been done have not shown better outcomes yet for those paired patients with drugs. So as of yet, this guy was pretty pessimistic, but in his view, we haven't gotten the precision oncology yet. However, I think that there's good hope to get there. So why is it so hard? We tend to have this idea. We know that each gene in our DNA is sort of a blueprint for a particular protein. All right, cool. So we have this idea that maybe we can send a cancer cell off, get it sequenced, and we can come back, get that result back, and we can look at it and we can say, oh, this gene has a mutation, it's broken, there's the culprit. That's not the case, and we tend to think that for a couple of reasons. One is that we oftentimes have our attention drawn to these cases where, for example, in breast cancer, the BRCA genes that we've all heard of, if there's a specific mutation in these genes, then you know that it really drives that cancer, or the probability that that cancer might occur. We hear about rare genetic mutations all the time that cause a certain type of muscular dystrophy. But in reality, most things require sort of a constellation of genetic defects to happen, and cancer is certainly one of those things. In the human genome, there are 3.2 billion base pairs, 3.2 billion letters in a 4-letter alphabet that occur. Of those 3.2 billion, there are about 1/100th of those base pairs that actually code for proteins, that are blueprints for proteins. Now, previously, people used to call the other remaining 99% of that data junk DNA, and I don't think people use that anymore. I think scientists and researchers and doctors are starting to realize that there is information there that probably is very relevant. However, right now, certainly in clinical settings, those 20,000 genes, that 1/100th is what folks tend to think of, what folks tend to focus on. And to give you some context, if someone goes to an oncologist and they have cancer and it's determined that they should get genetic testing done to help them with that cancer, typically that test is a test on a small panel of 50 genes out of these 20,000 genes, or if they really want to splurge, maybe it's a test on 200 genes. It's rarely on all 20,000, and it's never on all 3.2 billion base pairs. That's a good question. I don't know I thought Steve Jobs did some spiritual meditation. I don't know. I'll Google it later, though. It's a good question. He may have. But it's really interesting because there's a lot of information there. If you take those 20,000 genes and you get those sequenced and you figure out where the mutations are, you will typically find between hundreds and thousands of mutations. So thousands of things that are wrong in those genes. One of the oncologists we talked about in the customer discovery, we talked to in this customer discovery project for this particular project said, when you get the mutations back from somebody who's been sequenced, it looks like a truck has run over that person's genome. There's just all sorts of bad stuff that's happened in there. So it's really difficult to take this next step and say, okay, which of those 1,000 mutations really matter, which ones are driving this cancer, and which ones really wouldn't make any difference at all in terms of this person's health. And that's the tricky bit that we're going to talk about. Once you know that, once you can get past that, which ones matter, then you can say, okay, which ones of those mutations actually have some sort of action that can be taken, a drug, a therapy, something. But until you know which ones of those things matter, you don't have a way to figure that out. So obviously, patients, doctors are going to be pretty happy, but I think also researchers are going to be pretty happy to be able to really focus on which things drive a particular cancer often that they can focus on and really try to build drugs for. So the big questions are, and these are really some of the big unanswered questions in bioinformatics, which mutations are drivers of a particular cancer, not just broadly, but for your cancer, a particular patient's cancer, what matters when you get that information back? And the other side of that equation is how much do those drivers look like mutations that we know we can do something with, that there's some literature on, that there's a therapy for, that there's something that can be done for it? Those are the two big questions, and those questions have not really been answered yet. Right now, one of the older ways of finding cancer drivers is you take a whole bunch of tumor cells from a lot of different tumors for a particular type of cancer, and you look at the mutations in those cancers. And you say, okay, which of these mutations are occurring more in these cancers than this normal background rate for a normal healthy cell? All right, well, that's got a number of issues. First of all, it's very imprecise. It's really hard to figure out what normal is in the first place. And even if you do figure that out, you have no information about what mutations in a specific cancer really drive that particular cancer. You just have this broad data that you're not able to really utilize for a particular patient. So you can say, okay, usually you have this mutation. We see that often in this type of cancer. Maybe that's got something to do with this type of cancer. Not a very good way to do it. Now, it would be cool if somebody figured out a better way. So enter our protagonists. We have a team from the University of Pittsburgh that we got introduced to in the Department of Biomedical Informatics that came up with an algorithm that they call tumor-specific driver identification. So TDI for short. It's not a catchy name, but I think they're working on that. But it really tells you what they're trying to do. They had, at the point that we met them, a really functional research system where they could take information in. I'll tell you about how it works in a second. And they could spit out at the end of this process of this algorithm information about what particular mutations drive the cancer in a particular tumor. So they were at a research phase. They needed to go from there to a implementation phase. They want to try to sell it. They want to take it to initial partners or clients. So in steps the Pittsburgh Health Data Alliance, which is a group of, or a collaboration between UPMC Enterprises and two universities you may have heard of, Cardini and Pitt. And their idea is to take data and turn it into improved human health. And this was one of their, so these folks were one of their first projects. So they wanted to take that and really sort of figure out how to commercialize it. So very early in the process, it's a research-based system. How do we take this? Who do we take it to? Questions were very preliminary when we started working on this as software developers, and to some degree, innovators or helpers of innovators in doing things like determining who initial clients should be. So we worked on this project, and the thing that we started working with was this algorithm. So their algorithm took into account a few different things. Mutations from a specific cancer, not a group of cancers, but one particular cancer. And they take expression data. So if those mutations spell out a blueprint for proteins, they take data on what gets created by that blueprint within that cancer. And then they take data from a whole lot of tumors. So they take data from, I think, 5,000 tumors right now and use it as sort of training data. And they're able to do some things aside from, first of all, giving information about specific drivers for that tumor, but they're also able to tell you information about how one mutation looks in comparison to another. So if you have a mutation that nobody's ever seen before, they can tell you a little something about how similar it is to something that has been seen before. So their algorithm takes training data, takes cancer tumor data in, spits out drivers, but it has some other components. You've got to take raw data. A human has to figure out how to turn that raw data into something that this algorithm can deal with. And usually a graduate student is going to run a report for you from a SQL database. So it's a very research system. It's great, but it's not something that's ready for production yet. So I think the interesting part of this example is there are really a few things that have to happen in a big data system. The first one is the one that we think about often, which is how do we make it scalable? How do we make it repeatable? How do we make the data pipeline actually work? And we'll talk in a second about the other two, which I think are equally important. But to answer that first question, we replaced the graduate. No graduate students were hurt in the project, but we replaced the graduate student with a data pre-processing system. And we built some reports, even though we still didn't know at this point exactly who that final client was going to be. And then we really built a system to make it easy to get data in and to make it easy to store data that results from it. We put it into a scalable system. We used the Google Cloud Platform so that you could run 50 of these things at the same time. Each one of these runs takes 10 minutes. As they expand the data that they're going to collect, maybe each one of those runs is going to take an hour. So it's important to figure out how to scale that thing up. And we use some technologies that if anybody wants to geek out with me afterwards at the cocktail, I like to drink wine and talk about Docker. Please see me afterwards. But some really cool technologies to take a whole application and turn it into a container. Kubernetes is a platform to take containers and kind of orchestrate them and do auto scaling and do all that kind of good stuff that's necessary when you have this much data. And if anybody's interested, I think the Children's Illustrated Guide to Kubernetes is a very hilarious and great introduction to how this stuff works, but I won't talk about it now. I think the other interesting part of this is that a user interface for a system like this needs to be built not just for humans, but it's got to be built for computers. So an application programming interface, typically a company that's going to use something like this is going to have their computer system grab data from this computer system and display it in their own system. But when you're out there trying to sell it to your first initial partners or work with your first initial partners, you have to have a visual user interface. So we spent a lot of time making sure that we had an interface that was easy to use, that would let people, researchers, clinicians, put the data into the system and get a nice report out of the system. And then I think the final system that really mattered and always is going to matter, I think this is what some of the presenters this morning were talking about, is how do you make this data useful so that somebody can actually figure out what to take away from it. So we spent a lot of time working with the researchers, and they're the ones who guided us, but we, I think, helped a bit in structuring how this came out. We ended up categorizing drivers in terms of drivers that we already knew about, drivers that were novel in a way, and drivers that were just completely unknown. And we built a report around that that was visual in a sense, that showed how all those things worked, but I think really most interestingly showed how an unknown driver relates to drivers that have information out there that's already known about them, how similar these things are. So our hope is we can take this to an oncologist or a research partner, and they can look at it and see some value immediately without having to think too hard about how they would use this. And really, I think I'm out of time, and I think that's it. Thank you all for staying awake, and see you all during the cocktail hour. If anybody has any questions, please grab me afterwards, and we'll talk then. Thank you.